chemotaxis. Remember those two important steps where the neutrophils from the blood vessel have to migrate to the site of infection. So they must first attach to the endothelial cells, then do the diabetes or wriggle out through those endothelial cells, and then migrate to the site of infection, which we call as chemotaxis. But opsonization is the term that we use where the microbe is coated by antibody or complement or by both. And that process enhances phagocytosis. Okay? So remember those terms.
Dr. Reddy,
Question?
in terms of your complement activation, in lecture we mainly stressed on the 3, but your notes say there's 4 with the terminal pathway.
Oh, yes.
How deep do you want us to go into that?
Okay. The first 3 steps are, we said, classical pathway, for example. If an animal has seen this parasite before, means it has antibodies in its blood. So if the same infection occurs again, second time, these antibodies can bind to that microbe and activate complement proteins. So that's classical pathway. That requires antibodies. Okay, but later we found the alternate pathway where we don't need antibodies. So if there is a new pathogen coming into picture, animal doesn't have antibodies, but sometimes they can also induce directly in the absence of antibodies. That's alternate pathway. But we said more recently they found another pathway called as lectin pathway, where some lectins present in the blood of humans and animals can also bind to the microbe and activate complement. So that's a lectin pathway. Okay? It's similar kind of to that of antibody-mediated classical pathway, but here these are the lectins, are collagenous sugar-like proteins present in the blood that can activate pathway. So those are the initial pathways. Once these are initiated, there is a series of steps that leads to final terminal pathway, where that results in the production of membrane attack complex. Okay, so that's the terminal pathway.
So they all lead into the terminal
Yes,
pathway?
all these 3 pathways will lead to the terminal pathway. In that, some intermediary products like I will not ask, maybe I don't have any questions. Some components of that complement, like C5A or C3A, C3B, these are some of the intermediary products before the formation of the membrane attack complex. But like C3B can also participate in opsonization. We said complement can coat the bacteria, but C3B is one of the complement proteins. So it can coat the bacteria. So that's opsonization. And the process of chemotaxis, how does the neutrophil know to come out and migrate to the site of infection? Some of these complement proteins can do that, attract the neutrophils to the site of infection. Say, for example, C5A is one of the intermediary products that can attract neutrophils to the site of infection. Okay. That's about interference. Question?
I had a question about the pathogen-associated molecular patterns. Why is it that neutrophils don't have the surface receptors for those?
Pathogen-associated molecular patterns. Those are meant for the antigen-presenting cells to recognize those. neutrophils only do phagocytosis, okay? Whereas the antigen-presenting cells, such as dendritic cells and macrophages, have those pattern recognition receptors, or the PRRs, to recognize those molecular patterns. And once they bind each other, then they produce some cytokines. That process initiates the specific acquired immunity. Again, it depends on whether it's a virus or a bacteria or a fungus, and we said depending on that, they produce some specific cytokines that induces a specific acquired immunity that's required to specifically attack that virus. Again, different strokes, remember that. Okay. Again, interferons. Remember those three major kinds, alpha, beta, gamma. What are interferons doing? An interferon is produced by an infected cell, and that interferon will bind to the neighboring uninfected cell and make it resistant for viral infection. Okay? And in the process, we have three different kinds, alpha, beta, and gamma, which cells produce which we should be able to say. Okay. Eosinophils, anti-parasitic immunity, they have those granules. What do they have? Eosinophilic cationic proteins, major basic proteins, and eosinophilic neurotoxins or something like that. Anti-parasitic immunity. Mast cells, what do they produce? Histamine. Major component of those granules is histamine, but there are other things that will also be coming into picture. So those are the innate immune responses. Then comes the specific acquired immunity, which is mediated, again remember, it's mediated by T lymphocytes and B lymphocytes. Anything with the lymphocytes is specific acquired immunity. All other immune responses are innate. So if I ask, is cytotoxic T cell activity, which one is that? Is it innate immunity or specific acquired immunity? So you should remember that. If I ask IgM-mediated virus neutralization, for example, then it's specific acquired immunity. And if I ask eosinophil attacking a parasite, that's innate, that's not specific acquired immunity. Okay? And then coming to specific acquired immunity, we are only including in this block 1 mediated by B lymphocytes. Okay? Again, remember B lymphocytes differentiate into plasma cells, and they are the ones that are producing those 5 different antibody molecules. And you should be able to remember that basic structure of the immunoglobulin: 2 heavy chains, 2 light chains, and the heavy chain has that variable region, and then constant region that has CH1, CH2, and CH3. And we said there is a hinge region that's kind of in between CH1 and CH2. And then CH2 and CH3, we said major things to remember are where exactly they bind to the complement, or complement binds to which one, CH2 or CH3, or the FC receptors on phagocytic cells, where do they bind? Is it CH3 or CH2? You should remember that. Coming to the 5 immunoglobulins, then you should remember IgM is a very big molecule. It's a pentamer. It has 5 basic units. and it has that J chain binding those 5 basic units. And so, again, you might see a question, which antibody molecule has, say for example, 4 antigen binding sites? Or which immunoglobulin molecule has 10 antigen binding sites? Or which antibody molecule has J chain? J chains are present both on IgM and IgA, so remember that. And, which antibody molecule is produced first in the immune response to a new pathogen? It should be IgM. Okay. But then if I ask, if you give a second booster vaccine, which immunoglobulin will be predominant? IgG. What's the function of IgA? Where does it act mainly? Mucosal surfaces, and it's a dimer. It has 4 antigen binding sites. It has a J chain, and it also has that secretory component. Remember that, okay? What's the function of the secretory component?
So it doesn't kill it. Protects it.
Right. Protects from proteolytic enzymes, but it also helps in the transportation. IgE is involved in allergic reactions. How does it do that? It triggers the mast cell. Once it binds to an allergen, it triggers the mast cell to release histamine. Okay? So, but IgE is also involved in anti-parasitic immunity. You should remember that. And lastly, we have that polyclonal and monoclonal gammopathies, talking about myeloma, multiple myeloma. Okay, what kind of shape in the graph you will see in case of monoclonal gammopathy?
Spike. Sharp spike.
The base will be narrow, but there will be a big spike. Okay. Normal is a bell-shaped curve, but in the polyclonal gammopathy, then the base will be broader. But in monoclonal gammopathy, there will be a big spike. So, where do you see these monoclonal gammopathies? Mostly it will be multiple myeloma. So, what is multiple myeloma? It's a cancer of those plasma cells producing IgM or IgG type antibodies. Okay? And in the case of multiple myeloma, you will see a lot of plasma cells in the bone marrow. If you take a biopsy of the long bone marrow and look under microscope, you will see a lot of plasma cells. That is an indication. Other than that, we said we can do serum electrophoresis and look at these gamma globulin fraction. Okay? And also, we showed a picture, a radiograph or X-ray of the long bone, showing those radiolucent areas where the bone is eroded because of the proliferation of the plasma cells, and that causes calcium to be released into the blood. So you can also confirm multiple myeloma looking at the X-ray picture of long bones. Okay? So again, remember, we can do, so how exactly we confirm multiple myeloma is looking at the graph after the serum electrophoresis, and then look at the radiograph, And what else can we do? You can do biopsy of the long bone and look at a number of plasma cells. But I also said about Benz-Jones proteins, right? These are light chains of those immunoglobulin molecules. Plasma cells, they are proliferating so much, they produce these unnecessary immunoglobulins, especially the light chains, and they are secreted in the urine. Okay? You will see those light chains in the urine sample, which we call them as Benz-Jones proteins. So that's also indicative of multiple myeloma. Okay? That's all I have, unless you have any questions.
Would you ask us like nitty-gritty questions, like what's inside the primary granule of a neutrophil, something like that?
So what does primary granules of neutrophils have?
In the primary. Okay.
Maybe I will ask you. I expect you to know, but I might ask. Primary granules have lysozyme, for example. So what's the function of the lysozyme? How does it act against the bacteria? How does lactoferrin act?
Binds to the iron.
Okay.
Okay.
Okay,
Thank you.
that's all.
Thank you. That's nice of him.
Thank you.
No need for that.
Oh, yes, there is. All right, don't worry.